All right. Good morning, everyone. I'm going to be talking for much more than 5 minutes, unfortunately for you. But today, I think we can go ahead and get started talking about heterogeneity. This was, I think, kind of something that basically everyone said they were interested in. And I think it's a really exciting topic because I think it's an area where cryo-EM really excels because we are collecting images of these individual proteins. We kind of have access to this heterogeneous the information about the heterogeneity of the sample. So it's an exciting part of cryo-EM, but it is also, I think, one of the biggest challenges in processing cryo-EM data, depending on the types of heterogeneity you have. So because I'm going to be saying the word heterogeneity about 1,000 times today, I thought it'd be good to have a quick overview of what heterogeneity is, right? So far, all of our samples have been one protein, or RNA, sorry, in basically one conformation, right? And in some cases, there's been a symmetry-related difference in the maps, right? Where we had, for instance, the calmodulin and TRPV5 in one of these 4 positions, but the particles themselves were all basically the same. It was just a kind of artifact of how we chose to align those images that caused the difference, right? Heterogeneity is kind of acknowledging the fact that the particle images themselves are actually different. And like we said yesterday, probably in reality, if you were able to kind of shrink yourself down to the sort of angstrom scale, you would say all these particles are different, right? We're talking about kind of detectable on the scale of cryo-EM difference. In some cases, the heterogeneity is marginal and it can be ignored, right? As we saw with the trip channels, that C-terminal domain was a little bit flexible, but for the most part, we could either ignore it or just make a mask, right? Do some of these simpler solutions to kind of get a usable map. But in other cases, the map quality, either overall or in particular domains, will suffer until we handle this heterogeneity. I'm going to define a few types of heterogeneity, and I want to emphasize that this is kind of a categorization that I have found useful. I'm not claiming that this is exhaustive, nor am I claiming that no one would disagree with me. I'm sure there's a lot of ways you could categorize heterogeneity, and the way you categorize it might depend on what you're trying to do. But I'm going to talk about 3 kind of categories today. So one is kind of sample identity heterogeneity, which is where you're looking at images of just totally different things. I think a lot of people wouldn't even call this heterogeneity. That's just different stuff, right? So for instance, junk versus sample, that you could consider heterogeneity, or maybe sample A versus sample B in the same kind of images is also heterogeneity. Those you can usually resolve during particle curation, right? The particles are going to be so different, no worries, but that'll cost you a grid puck. The samples are so different that when you're doing 2D classification or ab initio or something, you'll be able to pull those apart. So those are a little easier to tackle, whereas some other types of heterogeneity, like compositional heterogeneity, might be harder, right? So compositional heterogeneity is saying the composition, right, the things that make up the particle are actually different. So maybe you have some kind of binder that is either present or absent, or a drug that is bound or unbound, right? So that would be compositional heterogeneity, but that's discrete. It's either there or it's not there. Finally, you have conformational heterogeneity, okay? And this is basically the entire protein is the same in terms of the atoms that are there, but they're in a different arrangement, they're in a different shape, okay? And this could be discrete or continuous, right? In reality, everything's continuous, but we can kind of treat them as discrete or continuous. You might have a protein that's either open or closed, and the transition between those 2 states is so fast that you'll have very few particles that are in an intermediate state, and so you can treat it as if it's either open or closed. Or you might have a situation like we saw in one of the lightning talks yesterday, where you have a fab bound to some domain that's very flexible, right? That would be more continuous conformational heterogeneity, also known as flexibility, right? Where you have this kind of continuous gradient where you have limits, right? There's either up or down, but you kind of are sampling that whole range in between. Okay? We're going to be using the example today to talk about heterogeneity for both sessions of an ion channel called FaNaC. If you can't tell, I love ion channels. FaNaC is the FMRF amide-gated sodium channel, so I'll just be saying FaNaC. And it's a member of an important family of ion channels. It's in a bunch of different parts of the tree of life. We're going to be using 2 different Empire data sets kind of interchangeably, 11631 and 11632. both of these movies come from FaNaC particles. The gene is from this worm, this ocean worm, and I'm sure it's a very important part of the kind of oceanic ecology, but I found it very gross to look at, so that's why there's gummy worms in the background. But the important thing to know is that this is an ion channel and it's ligand gated, okay? This is what the ion channel looks like, and I'm gonna belabor this point a little bit because when we're talking about flexibility, I'm gonna be talking about these domains a lot. So we actually know, the authors knew about this channel or a related channel ahead of time, just based on some sibling channels that we already had structures for. I'm showing you the model that these authors produced from this data. So we're actually looking at the FaNaC ion channel here, and I'm just gonna emphasize some important domains here. So just like with the trip channels, we have a transmembrane domain. That's down, if I can find my little button, that's down here. So these helices form the transmembrane domain that goes through the membrane. There's a pore for the sodium ions to go through. The other important domains are the thumb domain. That's this kind of outer part that's at the edge of the channel, and the finger domain. This field likes to think of the monomers. This is a homotrimer. The monomers is kind of a finger or a hand that's facing outward from the channel. And what happens is that thumb domain, right, this part here that is on the kind of outside of the channel, that'll pinch closed when the ligand is bound, and that will pull the TMD open, okay? So this is where the ligand binds. We're kind of here between the finger and thumb there. The ligand is FMRF amide, so it's basically four amino acids. So it's a pretty small ligand. You'll see we will actually be able to see this in the cryo-EM maps, which is nice because this data is really beautiful. But yeah, so it binds here. That's the ligand there. And then here's the thumb here and the finger here, right? So it really binds at the interface between these two domains. Okay, the last thing I'll show you is a morph that didn't work, of course. So you'll have to imagine it. But basically that thumb and finger just pinch together continuously. Okay, so that's what we're gonna kind of be trying to figure out, right? These authors wanna know, you know, what are the conformational changes that are associated with ligand binding? That's kind of always the question with these ion channels. When you're first investigating the effects of the ligand is like, which residues are involved? How is that change transferred all the way down to the other end of the protein, kind of the business end, the pore? Okay, so that means our goal is to resolve high quality maps of FaNaC1 in the apo, that means no ligand, and FMRFA or ligand bound states. All right. So this is actually the first data set that I'm showing that was deposited as movies. So as part of the standard workflow, which again, I'm going to kind of elide here, we did have to do patch motion correction. It basically works out of the box. The fun thing here I wanted to highlight is this is a visualization of the patches of one of the movies. So we're looking at, once we've done that rigid motion correction of the frames, we're now correcting the patches of the movie for the ice bowing, right? This kind of non-rigid motion. And you can see there's this little cluster of patches on the left-hand side that moves a bit more than the others. And that happens to overlap nicely with this contaminant there, which is often what you see. You don't need to do anything about this. It's just a fun thing to notice that we're kind of able to model the difference in movement of these different patches. Again, I do CTF estimation, and I use that fit quality to kind of throw away some micrographs that might be worse. But this is the first part of the standard workflow that I didn't mention before, because this is A, relatively new in CryoSPARC, and B, it requires movies. But this is the micrograph denoiser. And I really recommend, since you're always going to be starting from movies, I really recommend adding this to be part of your standard workflow, because we have found it makes a huge, huge difference in the quality of particles you pick from the micrographs. Okay, so here is a kind of representative micrograph from this data set. I would say this is actually a really nice micrograph. Here we can see this looks like a micelle side view. And then if you look, there's some little triangle-y shaped things that are the top view, right? This is a homotrimer, so it makes these little triangles. Here is the denoised micrograph, okay? This job is really, really fast. I think this ran in about 15 minutes for all the movies. And you get these really, really high contrast micrographs. And I want to flag here that the contrast here has been added, right? This is using a machine learning technique to kind of add contrast here. So we're not like discovering some contrast that's there, right? We're adding contrast to help us pick particles, but that's useful, right? We can pick particles on these denoised micrographs and then extract them from the real micrographs. And I think you shouldn't use a tool like this without knowing how it works. So I'm going to give you a quick kind of demonstration of how it works. What do you think this is an image of? Yeah, why would you know, right? If I show you another image and I say these are both images of the same thing, and I guarantee you these are both images of the same thing, now, if you're familiar with the Structura logo, you might say, that's the Structura logo. And the reason you're able to do that when you have two images that you know are the same thing is because when you only have the one image, right, this hole in the middle here, you don't know whether that's noise or whether that's really there in the object, okay? But if I show you two things and I tell you these are the same thing, right, anything that's in both of these, you can be much more confident that it's really there. Okay, that's basically a simplification of how this denoiser works. If you're interested in reading more, we have a lot on the guide page about it, or you can read about noise-to-noise training, which is how this machine learning algorithm works. But basically, we take the movies and split them into half movies, and then do that comparison, right? Anything that's in both of the half movies we know is real. Okay, so again, what I do is I pick images, I pick particles on the denoised micrograph because the contrast is so much higher. But that contrast isn't real, so I extract them from the real movies that have not been denoised. And CryoSPARC handles all of this for you. You just plug in the denoised movies, and it knows which ones you want to pick on and which ones you want to extract on. So there's no worry about setting a parameter wrong or something. Okay, so I just wanted to show that because we did do that for this data set, and it makes a huge difference just in general, but especially we have found it helps with preferred orientation. We had a question yesterday about how when you have orientation bias, you don't know whether that's because your particles are all in one orientation, or maybe one of those orientations is much easier to see and much easier to pick than the other orientations. And so you kind of have introduced orientation bias when you isn't really there. We have found that on some data sets, the denoiser alone can recover a lot of your missing views just because you can actually finally see those views to pick them. Okay. Here's the templates I made. So you can see we have a nice range of views from top through oblique all the way to side views. And then here is our consensus non-uniform refinement, right? That's pretty easy, and it's a pretty good looking map. So I'm going to go ahead and open it in ChimeraX eventually. Oh, come on. There we go. Yeah, so I colored it. That's why it was taking so long. So here I've colored the TMD, right? Thumb is here, and then the finger is up here along with the kind of scaffoldy part of the protein. And you can see we have, it's a really gorgeous map, right? We can see maybe some lipids here or some detergents, who knows. The transmembrane helices look really good, and the handedness is right? Up here, the finger looks pretty good. It's a little blurry there, but like back in here, you can see a lot of nice like side chain density and things. However, the thumb is much, much blurrier, right? We have pretty low quality map on the thumb. And this is, if you remember during the fundamentals, when we were talking about the GSFSC resolution, this map is actually sub 3 angstroms, GSFSC resolution. And this is why I really wanted to emphasize that the GSFSC, the global GSFSC, is just one number, right? On average, kind of in total, this is a very high resolution map. But if you look, one of the parts we care the most about, which is the thumb, right? We want to really learn how this thumb associates with the ligand and moves, is actually very poor resolution. Well, not very poor, but poor resolution, much poorer than 2.8, right? I wanted to highlight too that we can actually see the ligand in this consensus map. So I've made a little mesh around the ligand, and we can see really nice density following all those 4 amino acids. So it's a really high quality map, but we have this problem with the thumb, right? Remember these? Why do the thumb and the TMD, oh, the TMD is a little blurry on the bottom, I forgot to say that. Why do the thumb and TMD look blurry in this map? And also, another thing I forgot to show you, which I'll show you now. So we see this nice density around the ligand here, right? But as I contour the map down, watch the density around the ligand compared to the surrounding area, right? The ligand is disappearing faster than the finger in the background here, okay? As I contour the map there. So why does the thumb and TMD look a little bit blurry in this map? And why does the density for the ligand for FMRFA disappear at a lower contour than the density for the rest of the finger that's right next to it? Okay, so again, you'll have 1 minute to kind of think to yourself on these questions, and then 4 minutes to talk to a neighbor. Okay, so go ahead and start chatting with your neighbor about the blurry sections of this map and why you think that might be. Okay, so sounds like a lively conversation about the finger and thumb domain here. I think I heard a lot of people talking about flexibility, which is maybe there was a hint in the title of this part. But yes, the reason I think that we can't really see the thumb very well is flexibility. I think the reason we can't see the ligand very well is some combination of flexibility and partial occupancy, right? So these are important concepts when you're thinking about heterogeneity in general, right? So what we're doing here, right, is aligning all of the particle images to some static volume, right? Remember that forward model. We treat all the particles as if they come from one single static volume, okay? But we know the particles themselves, as they are either bound or unbound to this ligand, the thumb will be in position A or B, and maybe it'll be somewhere in between, right? But it'll either be an apo or ligand bound state, probably. So what's happening is we align all the particles to that rigid part, the part in the middle of the channel that doesn't change very much. And then some of the particles are bound to ligand and some are not. So when we back project the particles to make the 3D map, all of them contribute to that strong scaffold in the background where we get that really nice resolution. But the thumb is a little bit weaker than the surrounding density and it's kind of blurred out. And in addition, only the particles that have FMRFA there will add density to that FMRFA part of the map, right? So if only half of your particles have the ligand, the ligand will have only about half the strength. And then of course, if the ligand doesn't bind very tightly, it'll also move a little bit in the particle. So you'll see some blurring out there as well. Yeah, and then the same thing happens with the thumb, right? If my palm is always in the same position and my thumb is moving between these two, right? Those positions will kind of get smeared together, okay? So this, I think, based on what we've talked about so far and based on kind of my experience in cryo-EM, I'd say this is a perfect case for 3D classification. And the reason I think this is we have a really high quality, right? I told you sub 3 angstroms overall, a really high quality map of most of the channel, right? That means our data is good. And that means that our poses overall are probably pretty close to the right pose, right? Because we know this rigid part of the channel is aligned very well. So when we have good data and good poses, that makes me think 3D classification, because I don't need to mess around with the poses with something like heterogeneous refinement. I know the poses are good. I just want to classify into apo or ligand bound. Okay, so we're going to try a 3D classification job to separate apo and FMRFA bound particles. There's a few things to think about with the 3D classification parameters in this case, right? The first is, do we want to impose symmetry? I told you this is a homochimeric channel, so we could impose C3 symmetry, or we could choose not to, okay? Some advantages of not imposing C3 symmetry is that we don't know for sure that all three of the FMRFA binding positions are occupied by ligand. So if we impose symmetry, we are preventing, we're forcing the classes to be either all three bound or all three not bound, right? Or we're hoping that's what happens, or they might all get half bound, but hopefully that doesn't happen. So we wouldn't be able to see if there's like one bound and two not bound, vice versa. However, to fully capture the combinatorics of those three binding sites, we would actually need 8 classes, right? And this is a big thing with symmetry where even though, to me, right, knowing what this channel is, it's the same if the position at the top left has ligand and the other two don't, as if the position in the top right has ligand and the other two don't, right? I know that those are symmetrically related. There's no way for the 3D classification algorithm to know that those are not meaningful differences, right? So it needs a class to put those two into. We could solve this problem by using symmetry expansion, which basically creates additional copies of the ligand, and then classifying on just one monomer, right? That's one way to work around this. But let's just say, for the sake of not spending a bunch of time here together, that we don't want to do that, okay? So we just want to do 3D classification. So we would need 8 classes to properly capture that, but we could do it if we don't want to impose symmetry. If we do impose symmetry, we would have more signal-to-noise ratio, right? Assuming that most of the channels either have 3 positions bound or 3 positions unbound, that this intermediate stage where some of the binding regions have ligand and some don't is small, then we could maybe say imposing symmetry is mostly right, and then we'd have much more SNR to help our classification move forward, right? So that's what I kind of think about when I think about whether or not I want to impose symmetry. Hard classification, right? We saw that if you turn on hard classification, right, you're forcing the particles to either be 100% in class A or 100% in class B. I either have ligand bound or I don't, right? That can help to separate things that look very similar. And we expect these classes to be very, very similar, right? Not only will they have the same rigid domain, but actually they have the same thumb, just shifted a little bit, right? So we expect these classes to be very similar. A particle that belongs to the ligand bound class will still look probably 80-ish% like the apo class, okay? So maybe hard classification is good. However, we're looking at a ligand, right? And there's a associated change in the thumb domain when the ligand binds, but the ligand is very small. So we're probably gonna want a small filter resolution, right? Remember yesterday when we talked about 3D classification, filter resolution is this thing that sets kind of the scale of difference that we're looking for. Ligand binding is a pretty small difference, and so we're gonna need a small, relatively small filter resolution. And we saw that with a small filter resolution, meaning high resolution, unfortunately, backwards, you know, the hard classification can collapse all the particles into a single good class and then put like 100 bad particles into the other class. So that's another thing to think about. Do we want hard classification or no? The last thing to think about, speaking of filter resolution, is what filter resolution should we choose, right? If we go too low, we may not be able to see the thumb and FMRFA, whether they're there or not, right? We're talking about a movement on the scale of a few angstroms. If we go too high, we might have that problem we saw yesterday, where all the particles end up in a good class, and then there's this really small bad class. So basically, the job doesn't do anything, okay? I know it's 2 in a row, but I think this will be fun. How would you set these parameters, right? What do you think you would do? What would be your first job or jobs that you'd set up thinking about these problems? And, you know, of course, it's totally fine to say, and I would probably encourage you to say, I would try this job with these settings and this job with these settings, because, you know, that's what I actually do when I don't just sit there and launch one job and then watch it and see how it goes, right? So you're going to launch a couple. Which ones would you set up? What parameters would you set? And why would you set them that way? So again, you'll have 1 minute to kind of think through what you might set up, and then 4 minutes to talk with your neighbor. All right, so why don't you go ahead and chat with your neighbor about what 3D classification jobs you'd set up and why. Awesome. So I'm sure that you all came up with great ideas for the jobs to set up. And I just want to emphasize that there is certainly no right answer to these questions. I think basically any combination of trying this on and this off and this filter resolution and that one would be right in the sense that you learn something about the sample, right? So I'm going to show you the jobs that I set up, but know that I actually, if you look at the workspace, I set up like 15 jobs and I'm showing you kind of the interesting ones because it really is, I think, something that I think part of the fun of CryoEM is you can really explore what happens looking at different things, right? Prioritizing maybe, you know, this high resolution stuff versus low resolution stuff, right? So I think you should always, I think the reason I wanted you to do this question kind of 2 in a row is that this is really how I recommend you approach these problems. I recommend before you even open the job, sit down and think to yourself, all right, what are the particles doing here? And what kind of levers do I have to investigate the different facets of how these particle images are different, right? And then you can think about, okay, if I set this one this way, I'll be ignoring this and prioritizing this. If I set it this way, it'll be vice versa. Then set up both of those jobs, right? So I think that's easier to do before you even open the job, right? Just think, what are the particles doing? And then you'll have kind of that foundation to approach the job itself. So here's the job that I set up. I'm not going to use a focus mask here. I'm just going to use a solvent mask from the consensus non-uniform refinement. So I'm kind of just saying classify the whole protein, okay? So yeah, no focus mask. I decided not to impose symmetry just because let's just see what comes out. But I'm still going to use 2 classes. So I'm kind of not following the combinatoric advice of, oh, then you need 8 classes. I don't know. Let's just try to see what happens, right? For filter resolution and the initial low pass, I set both to 6 angstroms. To me, that kind of just felt right. You know, I in honesty, tried everywhere from 4 through like 10 angstroms just to see what worked. I'm just going to show you 6 here. Again, I use the initialization mode of PCA, which, as a reminder, you actually do a bunch of little reconstructions and then kind of try to find the sort of groups that are most similar to each other and then sort of take the average of those groups. So it's a way of kind of trying to get your initial volumes to start very different. And that's because, you know, I know that these things are going to be similar, so I want my initial volumes to be quite different. And finally, I chose to set force hard classification true. A filter resolution of 6 angstroms is probably blurry enough to avoid that class collapse problem. And of course, you can always come back and launch the same job with it turned off or increase the filter resolution, right? So just as a refresher, what's happening in this job is that the particle poses are fixed, okay? So we have this non-uniform refinement that I did as part of the standard workflow, quote unquote, where we got this nice map with this blurry thumb, okay? So we're using the poses from that. 2 volumes are initialized using PCA, so we do all those reconstructions and find kind of the average. Particles are iteratively assigned to the volume that they match best, right? So the volumes change only when the particles change class. And here's the result. If you look on the left, I'm showing you kind of an overview of the whole channel, and then I highlight the region we've zoomed into on the right. Throughout, I'm going to try and be as consistent as I can with coloring the apo map in red and the ligand-bound map in yellow, okay? So if you look at the overview, it looks like we actually did a pretty good job, right? If you look at the thumb domain, which is on kind of the far left side of the ECD, it is pulled kind of out and away from the center of the channel. So we have successfully done some kind of classification without realigning these particles that separated out thumb over here versus thumb over here, right? But in my little slider here, if we compare the 2 maps, okay, the yellow map on the right, here's the density for FMRFA. This is where that ligand binds. You can see we still have really nice ligand density, which makes sense. That's the ligand-bound map. And the thumb looks much, much better, right? If you remember our consensus refinement, the thumb had this kind of blunted, sort of flattened out topology, especially on this side where we know it moves down when it's not bound to the ligand. So our ligand-bound map looks much, much, much better. If we compare that with the apo map, there's something weird going on, right? First of all, the thumb has moved, right? If we look at the overview, the thumb is in a different place in our apo class than in our ligand-bound class. However, it looks like garbage, right? It's still really blurry. We're not seeing that nice side chain density that we see for the ligand-bound map. Okay, that's one weird thing. Yeah, like what is this? Who knows? The other weird thing is that this is where FMRF would be if it were there, right? It's certainly weaker, right? It's kind of missing a little bit. but it still sort of seems to be there, right? If I sort of do this comparison here, you can see it's much stronger in the ligand-bound map, but it is still there in the apo class, right? And remember, these are just classes, right? So it seems like we were maybe partially successful, right? Maybe the ligand class was really did a good job. We pulled all those ligand particles into one class, but maybe our so-called apo class still had some ligand in it, right? So we didn't fully separate out ligand and apo. So I'm going to use the exact same parameters for another 3D classification job, but I'm going to add a focus mask. It says solvent mask there. I'm adding a focus mask using the same solvent mask, but adding a focus mask that's just on the ligand binding region. And to refresh your memory, what the focus mask does is it says, ignore all the variability that is outside of the focus mask, right? So the focus mask is what changes every time a particle switches class. What's between the focus mask and the solvent mask is the same in all the classes, and what's outside the solvent mask is 0, okay? Question? Oh, yes, good question. So I didn't, no, that's fine. That's a really good question. Let's just look. They look the same, to answer your question. Yeah, but we can just, oh, I forgot I even did this. Well, I'm glad you asked because I forgot I had a nice little thing. So we can actually, I made a morph here. So here's our ligand bound state. And we can see, so a morph in ChimeraX, right? We're not actually solving maps between, we're just interpolating between one map and the other. I made this just to show we are seeing, there is like some kind of movement here, right? Between these two maps. And what's nice is if you look out at the whole ECD, right? So we're looking at the top down of the channel now. We're not just, because we didn't use a focus mask, we were able to classify the whole ECD. You can see the whole thing is having this kind of twisting, rearranging movement that we expect to see. So that's really nice. Then for the question of what does the three positions look like, they all look basically the same. So here's this ligand with this blurry thumb. Here's this kind of ligand that we think shouldn't be there, right? Yeah, no, it's a good question because we didn't impose symmetry. So it's good to check all of your symmetric places. Maybe only one had this partial classification problem. Yeah. Okay. So now we'll make a 3D classification job where we focus on those 3 ligand binding regions. So this is what my focus mask looks like. I basically made just spheres around that interface between the finger and the thumb. Okay, and it's okay that there's these 3 disconnected blobs, right? Because it's all in that solvent mask. So what's between the ligand binding regions and like the bottom of the ECD and the TMD, that will be the same in both classes, right? And we're forcing it to be the same, right? Whereas in the focus mask, we're basically just saying, are the finger and thumb closer together or further apart? Okay, they look the same, right? So here is with just the solvent mask, here is with the solvent mask and a focus mask. You can see the red map still has this kind of fake ligand. It basically looks the same as it does over here. And in fact, when I was making this slideshow, I was so sure that I had just put the wrong images in here that I had to ask my partner to read the text to me. But the 2 jobs found apo particles that were 96% identical. Okay, so that focus mask didn't really change how much we were classifying things. And what that says to me is it could be any number of things. The 2 things that come to mind are, we know that the finger and thumb are pinching together, right? And we've made a focus mask kind of around this part. We also saw in that morph that pinching together and apart is coupled with a kind of global rearrangement of the whole structure. And this makes sense because that ligand binding region, you have to transmit that signal all the way down to the TMD. So the whole channel is rearranging and maybe forcing the rest of the channel to be the same in both classes sort of weakened our ability to tell the 2 apart. Okay, that's one thing. The other thing is that we actually are classifying these particles fully, right? Maybe we're successfully teasing these things apart and there's just something else going on here. So we're definitely able to pull out these FMRFA bound particles, right? And I think this is true because our ligand bound map, right? The yellow map looks much better as a result of the 3D classification. So we're doing some kind of splitting into 2 sets where the yellow map looks much, much better, right? The thumb looks much sharper. Also, the red map does have kind of overall that shift of that thumb away from the center. So we are classifying somehow on the thumb. It's not just good and bad particles that make that yellow map better. We're pulling out particles that seem to have a thumb in a different place, but we're still seeing some leftover ligand density. The thumb is still kind of blurry, right? So if we assume, and we are assuming at this point, but if we assume that we're successfully classifying these particles, that our yellow map really is all of the ligand-bound particles, and our red map really is all of the apo particles, right? If we're successfully classifying them, why does the apo map look so much worse, especially in the thumb? And why does it still look like it has a little bit of ligand in it? And while you're thinking about this, I encourage you to consider where we got these particles poses from, right? Remember, we've only been doing 3D classification. So where do these particles poses come from? Okay? So again, you'll have 1 minute to think and then 4 minutes to discuss. Okay, so why don't you go ahead and discuss with your neighbor why you think the apo map might look so much worse than the ligand-bound map? All right. So we're trying to figure out why these maps, one looks so good and one looks so not good, right? To answer this question, let's imagine a scenario. If you look on the SLAC website, you need a little badge to come in here. And let's imagine that this morning, I pat my pockets and I go, oh my God, I forgot my badge and I have to be in there in 5 minutes to give my talk. That's okay. I found a yellow piece of paper. So I'll just say, yeah, here's my ID. Can I please come in and into this national lab and give my talk, right? All of the particles that we have, both the apo and the ligand-bound particles, had to kind of show an ID to get into that consensus refinement, right? They have to get aligned to the same volume, okay? And for whatever reason, our consensus volume had ligand, right? Remember I showed you in the beginning, we saw a really nice density for that FMRFA ligand. And what that means is when we're aligning the apo particles to that consensus map, if they have any noise at all in that region or near that region where that ligand is, they're going to try and put that noise there to say, oh, yeah, I've got ligand. It's right here. It's this yellow piece of paper, right? So what that means is our apo particles have been aligned to a ligand-bound map. They're also going to try and put their thumb into the ligand-bound position, right? It's okay if it blurs out the background a little bit. I'll put my thumb here and then I'll be let in, okay? So 3D classification is using those poses from the consensus refinement. So it can't get rid of that overfit ligand. And it's not significantly overfit, right? We saw it was barely there, and we saw that the ligand-bound map still has that ligand exactly where we expect. It's just these particles are in the wrong place, okay? But we're using their wrong poses that we got from that consensus refinement. If this is true, right? So far, we're assuming that our classification worked. There might be something else wrong here. But if it's true that the classification was successful, and it's just the poses that are wrong, if we just do a local refinement or even a global refinement, but if we just do a refinement of some kind to update those poses, that FMRFA in the apo map should go away and the thumb should sharpen up, right? Because all we need to do now, we've separated the particles, we just need to update the poses. So let's test that hypothesis. But just remember, it's always about poses, okay? I said during the fundamentals part, I think the poses of your particles are the most important thing. This is the thing you should always be thinking about because it's the hardest thing for us to figure out, right? All of these refinements, all of this stuff, the classification even, you could think about it as getting a group that will have similar poses relative to some volume, right? It's always about the poses. So it's good to always think about the poses, okay? So anyway, this local refinement job. I'm going to take just the apo class, that red map that had the bad thumb and that kind of weird half ligand. I'm going to take just those particles and that same map, okay? So we're refining them against that same map that has that fake ligand. I don't want to bias it and say, no, I think these particles don't have that ligand there, right? I'm going to use that same data. I'm going to use kind of the standard settings I have for the Gaussian. The only thing is I'm going to decrease the amount of shifts a little bit because I expect that it's just they've been rotated a small amount. So I'm going to shrink the shift search a little bit, but that's all. You could probably leave that default if you wanted. So what's happening in this job is we've taken only the particles from that putative apo map, right? The one we're saying, I think that I have successfully taken all the particles that don't have FMRFA, okay? I'm going to align all of these particles to that map with a blurry thumb and that small bit of FMRFA left over, okay? And I'm only letting them rotate and shift a little bit around where they were aligned in the consensus map. Okay, so we're mostly sticking to the poses we already found, because again, we think they're probably pretty good. Even this blurry apo map has a good kind of structure backbone, right? It's still pretty good resolution. It's just this thumb and ligand area that's a problem, okay? And the volume changes when the particles poses change, right? We're not doing any classification here, so it's just as we align those particles, the volume changes, and we iterate that, right? So on the left, in kind of faded red, is the volume from 3D classification. And on the right, in dark red, is the result of our local refinement. And I get to do the fun little reveal where I say, boom, it worked exactly as we expected, right? If you compare the thumb here, we have this blurring between these domains. There's all this fragmentation and noise here. We can't really see the side chains there. Suddenly, the helical pitch is very obvious, and these larger residues are clear. This loop is visible, and importantly, that FMRFA density is completely gone, right? I can kind of convince you that it really is gone. We can open this up. And even as I contour down, you know, it's okay. There's a little noise there. Maybe the classification wasn't quite complete, But if you compare this, right, we have to come all the way down to where we're basically looking at nothing to see even that slight little blurring, compared to earlier where it was this clear little bit of noise there that might have been ligands, right? So it turns out that we had successfully classified out our particles, but it was those leftover poses from when those particles were aligned to the ligand-bound state that were causing the map to be poor. Okay? So even though the poses looked good when they were together, the poses were the problem, right? So the poses were the thing that made us pick 3D classification in the first place, right? I told you, hey, I looked at this map. It looks pretty good. That means the poses are good. Let's not update them. Maybe I was wrong, right? Maybe that assumption was wrong. What if we had tried heterogeneous refinement in the first place, right? What if my whole thing was backwards, where I said the poses are good and it's too many degrees of freedom, right? So let's create a heterogeneous refinement job. I'm going to give 2 copies of the volume from the consensus refinement. And remember, this volume is the one that has a bit of a blurry thumb, but a strong ligand density, okay, and high quality kind of backbone of it. I'm going to force hard classification because I expect them to be different. And here I am going to impose C3 symmetry. And the reason I'm choosing to impose symmetry here is that because we're updating the poses, I want to make sure that my signal to noise ratio is as high as I can get. Okay, so because we're doing a lot more degrees of freedom here, right? So in this job, the particles are now going to be aligned to each of those 2 volumes every iteration. And since we gave identical volumes in the beginning, they're going to be forced to be kind of randomly assigned at first, but then as the iterations proceed and the volumes diverge, they'll be put into only whichever one they match better. So the volumes will change when particles change class or when their poses change, okay? Poses. Here's the result. Whoa, okay, there was some junk in the particle stack still. That might be another problem here, right? It looks like this green class is just kind of bad particles, maybe thicker ice, maybe starting to denature at the air-water interface, who knows? They're just bad. This is another advantage of using heterogeneous refinement, is that even if you kind of expect to see 2 classes similar, blah, 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 heterogeneous refinement can find this junk, okay? This junk class is actually almost a quarter of our particles. So that might be why we had a little bit of blurring in that apo map. That last little bit of fake density might just be noise that had been kind of aligned there, okay? So it turns out the particle stack wasn't actually fully clean. So we're just going to repeat that exact same job again, take only the good class from the previous heterogeneous refinement. And now we see what we kind of expected. I'm showing you the same thing here, where red is my kind of putative apo map, and yellow is the ligand-bound map. You can see, again, we see a nice movement of the thumb up there. But if we look down here, the yellow map has really nice FMRFA density here, and then the thumb looks really beautiful. And the same thing is true of our apo map, okay? So not only did heterogeneous refinement clear out some of our particles, all those junk particles that we actually don't want at all, it did a much better job of finding us our 2 classes because it was able to update those poses. It didn't have to rely on the poses from the consensus refinement, okay? So I showed you this really in the order I ran these jobs, and really kind of with that result of finding those junk particles I didn't even know were there, right? I'm not like making up a story for dramatic effect. This is actually how I thought was best to process this data, okay? So I'm telling you this because this is my intuition after doing cryo-EM for a lot of years, right? Is that, okay, I think 3D class is best here. It turns out I was wrong, right? Your intuition is useful. I think in general, I would still, this doesn't change my mind. In general, I would still reach for 3D classification in that case, but more broadly, I would try everything I could think of. And this is something you could print out and put on a poster with like a cat hanging onto a laundry line. You only know your best idea if you try all of them, right? If you think of all your ideas and then only try the one you think will work, you might have missed something, okay? So I really, really encourage you when you're processing your cryo-EM data to try just every single thing you can think of, because you'll slowly learn what just never works, right? A high filter resolution with force hard classification just never works. And eventually you'll just stop trying it. But you'll also learn, okay, sometimes this works and sometimes this works, so I always need to try both. Okay, and that's what I'm trying to show you with this kind of story, is that we have this discrete classification with very good poses in general, but in this case, the poses weren't quite good enough. So one last thing I want to show you here, and this is one that I may not have tried, but I just want to show it, is that we saw in other examples that ab initio reconstruction is pretty good at both separating out junk And sometimes it's separating out different poses or different orientations, right? So what if we had tried ab initio reconstruction here instead of heterogeneous reconstruction? Ab initio, for a lot of reasons, has much fewer levers to pull. So here I'm just going to set, we're going to use those same particles, the ones from the consensus refinement, so including the ones we know now there's junk in there. And I'm going to ask for 6 classes. The reason I'm asking for a bunch instead of just 2 is that ab initio is really good at pulling out junk. So if I just give 2, it would do what heterogeneous refinement did and give me 1 good, 1 junk. Probably if I asked for 3, it would find some other kind of junk, right? So I want to give it lots of room, basically, to show me a bunch of different classes. Here's the result. Okay, right? There's some stuff that looks like FaNaC. Certainly this red class here and this pink class here. Maybe blue and green look like just the ECD, and then these are junk. It's hard to tell, right? Ab initio is not really built to do this, right? The goal of ab initio is to find you volumes that are good enough to do something else, okay? If you take these volumes and then run them through heterogeneous refinement, it works perfectly, right? It does exactly what you'd want, but at that point, why not just run a heterogeneous refinement in the first place, okay? I'm just showing you this to kind of emphasize, in some cases, those 2 jobs can do very similar things, but they are really built to do sort of different things. They sort of operate in different domains, right? I think of ab initio as you're just like going as fast as you can to get something. Heterogeneous refinement is when you kind of are a little more methodical and trying to separate out 2 things based on a small feature or a small set of features. Okay. I know I'm kind of well ahead of well, maybe not well ahead. Oh, I am way ahead. Okay. Well, let's just do questions, I guess. And if you have any questions from yesterday as well, we can do those too. Yeah. Oh, we have to wait.
I have 2 questions.
Okay.
The first is, where are particles with only 1 or 2 copies of a ligand?
Probably.
Like where are they? Which class do they belong to?
Oh, where are they?
Yeah.
Probably in both. Because we only asked for 2. You know, there's no way to say. I think probably more of the particles with 2 ligands are in the ligand bound, and probably more of the particles with 1 ligand are in the apo. But you know, I wouldn't say that for sure. I think if you really were interested in figuring out the stoichiometry of binding, what you'd probably want to do is do symmetry expansion so that you could think about each monomer individually, right? Because these 3 monomers are identical. So if you do C3 symmetry expansion and then classify with a mask around just 1 of the monomers, you're really looking at all 3, but you only need to have 2 classes. Again, ligand bound or not bound. And then you can go back through, I talked about this a little bit during the questions yesterday, you can go back through with something like CryoSPARC tools or particle sets maybe, and kind of count how many times a particle is in one class or the other. And that would let you find particles that have 0, 1, 2, or 3 ligand bound. Again, at that point, you're kind of making 3 independent measurements on one very noisy image. So I think the question of like substoichiometric binding with something that relatively small is a little tricky, but that's how I would do it.
The other thing is regarding this very real SLAC ID, the synergy you use. It feels difficult to believe that the density of a small ligand can dominate the alignment. Because it seems to me when you do this non-uniform refinement or what at the very beginning, in my understanding, what dominant alignment is the transmembrane domain, which is quite consistent between all particles, and then to a less extent the thumb and finger, which gives you your initial poses. And then you already have very well resolved the transmembrane domain. But my question is, after you do the local refinement of your pose state, you update the poses, assuming you only have 1 single global minimum, that updated pose most likely will change the conformation of your transmembrane domain. Is that what you observe?
Yes, it is. So your question is basically, once we've separated them out and done the local refinement, we see the ligand-bound transmembrane domain. Is that?
Yeah. Yes.
Yeah, we do. I'm trying to make sure I have the. Yeah, I do. Here we go. So here's our two maps, and we can see, so our TMD is still rather blurry, and it's not very well resolved. I think the reason for this is there's also some continuous flexibility down in the TMD, and we'll see that in the next section, that actually the TMD itself is wobbling around like this, back and forth. And so we don't see it especially well in these maps. I think that if we wanted, we could make a mask locally refined focused on the TMD. There's so much more mass up here that I think basically we're aligning the top and letting the legs kind of swing back and forth.
Thanks.
Hi. So when you're doing these classifications, how much do you play with the class similarity value rather than hard classification, or is it always just filter resolution?
So that's a good question. The question being, how much do I lean on class similarity versus hard classification? So class similarity is a parameter we haven't really talked about yet, or really at all, which is, so in ab initio and 3D classification, there's something called class similarity, which is a way of sort of handling this problem of class collapse, where all the particles go to one class. So class similarity is we force particles to be kind of spread among the classes evenly according to the class similarity. So if you have two classes, A and B, and your class similarity is set to 0.5, you basically assign half of the particle's density to both classes equally, and then half to the one that it goes to, basically, right? So it's a way of saying, if you set class similarity to 1, all the particles contribute equally to all the classes. If you set class similarity to 0, particles only contribute according to their similarity to that class. So it's a way of kind of interpolating between not classifying at all, really, and classifying according to what the algorithm tells you. And you typically anneal that. So you start it at some relatively high value, maybe 0.8 or whatever, and then you'll anneal it to 0, so that at the end of the job, you're only putting particles in the class that you think they belong to. And this is helpful if you have like classes like this, which are very similar, if you also have junk in the data set. Because if you look here, right, what happened here is that we had junk in the particle stack. And so what happened is all of our good particles went into a single class, and then all the junk went into another class. We weren't able to separate out the good and bad particles because there was junk in the good class, or sorry, there was junk in the particle stack. If we had had a higher initial similarity, we would have forced those maps to stay similar longer, which might have encouraged more particles to go into both. So that's what class similarity is. How often do I use it? I use it fairly often in cases like that where I expect my classes to be similar. I find it, I don't have a good heuristic as to kind of what to set it to. There is the auto-tune parameter that will try and help you figure out. And Michael and Ollie might have more suggestions on how to set these parameters. But I find that if hard classification isn't working for me, I'll launch a couple jobs with different initial class similarities and see which one works best. I don't know if you guys have a better heuristic than that, or.
Yeah, I think one approach that I've used with the class similarity parameter is unless I'm looking at a very, very tiny difference, I'll typically keep it at 0.5. And then if I notice the issue of class collapse where like all the particles kind of go to one class and you end up with like 9 empty classes and one class with a bunch of particles, then I'll start to increase that a little bit just to help the algorithm stay a bit more stable. That's one approach that we sometimes use. Yeah.
Okay, my question is still on this slide. So when I miss it, what's your input volume? It's 2 copies of identical consensus.
Mm-hmm.
Okay, what's the 2? The yellow one is bound state or unbound state?
Yes, that's a good question. So the yellow map here, where we have this good volume and the bad volume, the yellow one is basically identical to the consensus, where it still has the ligand bound. Yeah.
So is pure, is separate away all the unbound state?
So this still will have, so the result of this next one, where we have the 2 states, is all the particles that were in that yellow map, the non-junk map there. So this map still has ligand bound and non-ligand bound particles in it. So it's basically all the particles that aren't junk. So we start with our consensus refinement, which has both apo particles and ligand-bound particles and junk particles. What this job did is it separated out the two, ligand-bound and apo, from the junk. And then the next job took ligand-bound and apo and made two separate maps of those.
Just one question. Before you did the first 3D classification with C1, the consensus before that was also from non-uniform, right? That was done with C3 or C1?
That was done with C3, yes. The initial consensus refinement I did with C3.
And even after the C3, you can still do a 3D classification with C1?
Yes, so that's a good point. What's nice about 3D classification is we don't provide an input volume, right? So even though we've imposed the C3 symmetry on the volume during non-uniform refinement, so the particles' poses don't have any preference for one orientation or the other, we're kind of regenerating new volumes for the 3D classification. We just don't impose symmetry on those. You know, if you think about the problems we had yesterday with like trip V5, where because we had imposed C4 symmetry, it was hard for particles to kind of break out of that. That's less of a problem with 3D classification because we're generating new volumes directly from the particle images that we never imposed symmetry on. And of course, if there was a big symmetry breaking feature and we had imposed symmetry during the consensus refinement, then their poses would have been aligned to a symmetric map. So they would still have like a symmetry problem, and it might be best to go back and find new poses, as we saw here, right? The poses ended up being the problem. So it might be best to go back and try and find new poses. Of course, the problem here wasn't symmetry. It was just the general problem of ligand and whatever. So yeah, your point, I think, is right, that it works better than you might think, but still it's probably best if you think there's a symmetry problem to always stay in C1.
Thanks. There's one more, sorry.
Yeah.
About the expansion. Let's say if we do expansion and we do the refinement of each single subunit, and we found out that there are certain number of subunit has the ligand, certain number of the subunit doesn't have it. Then what after that?
That's a good question. So I first want to make sure that I was clear that once you've expanded, if you classify on just one of the subunits, just subunit A, let's say, because you've done the symmetry expansion, you've also rotated all of B into that same mask. And you've also rotated all of C into that same mask, right? Because you created, in this case, those 3 copies and turned them all by 120 degrees. So you don't need to classify each subunit individually. When you just classify on one, you've actually classified all 3, okay? So now you have a list of particles, and each of those particles, you've checked all 3 orientations based on the symmetry expansion. Does it have ligand or not, right? So then let's say you go through, use CryoSPARC tools to count how many times is this particle in yes ligand and how many times no. So now you have, for every particle, this particle was in, it had 1 time it ended up in the yes ligand class and 2 times it ended up in the no ligand class. That tells you that 1 of the positions has ligand and 2 don't, theoretically, right? So now what you can do if you want is go back through and say, okay, take all the particles that have no ligand bound, that never went into the yes ligand class, put all those in a group, and then unsymmetry expand them, right? Just take one of those images. Then you can do the same thing with the particles that were only in the ligand group once. The same thing with the ligand particles that were only in the ligand group twice, etc. So now you have copies, again, you've gone back down to single images because you're throwing away the multiples of all these images. But now you have a single copy of particles that you think only have 0, 1, 2, or 3 copies of ligand bound to that particle. And then you can try either a global refinement or you can try and figure out what orientation, like if it has 1 ligand, is the ligand there, there, or there? That's a little more tricky, but you could try. I would generally say if you group them and then try a global refinement, that might work best. Or you could try a 3D classification, since you know there's only this group of particles only has 1 ligand. You could do a classification with 3 classes and see where that ligand ends up.
Some questions from the Zoom people.
Great.
Many of the questions yesterday referred to very similar things we're talking about here.
Great.
Well, yesterday when you're talking about something, well, why didn't you try 3D classification instead? Why didn't you try heterogeneous refinement? And you're answering that today by saying try them all. So this answers all those questions. Almost 50% of the questions from yesterday was, why didn't you try this in this case?
Yeah.
And your answer to all of them is try them all.
Right. Yeah.
Okay. And the second question that has come up today, and probably also yesterday, is your absolutely great use of Chimera in all of these things. How did you do that? What did you do? And to generalize the usefulness of Chimera for everyone in the cryo-EM community, you could shout it out to them.
Oh yeah, ChimeraX is probably my favorite piece of software on earth. We were talking yesterday about, I think the whole field owes a great deal to Tom Goddard. I don't really know what we'd do if we were still kind of banging rocks together and looking at, 2D printouts of particle images. But gosh, what do I do with ChimeraX is kind of a big question. If there's specific questions about like specific ways of looking at maps, I'd be happy to answer those. Yeah.
Sorry. The one was, let's see, how to create and label different domains in CryoEX. It looks really nice to have clickable domain labels in ChimeraX.
Oh yeah. Okay, so I can talk about that for sure. So what I do there, let's go back to, because I had the one yesterday too of trip. I think this has it. So to do this, there's a few things. So I actually here hidden have the atomic model, and I colored that based on the domains, which I just found the residue numbers and colored them. And then there's a command called color zone or surface. No, yeah, color zone. Anyway, it's like color zone near whatever. You can just search help color zone. Yeah, there it is. So you say color zone, and then you give your map, and then near your model, and it will color the map the color of the model that's closest to that surface, and you can give it some other parameters as well. So what I did here, if you look, is I colored the thumb orange, the TMD green, and then everything else is yellow, except for the FMRFA I colored pink. And so if we look at my map, we see basically the same thing. I've hidden the ligand, or maybe it's too low. But anyway, that's how I did that. To get these buttons, or like if we look at this part, this is called a button panel. So you can say like button panel, FaNaC1, add, I don't know, best and window size. And it'll add a little button there, and it'll run that command. So when I click this thumb command, it runs the command to create this surface, that kind of thing. When I'm using cryo-EM for myself, I don't generally do this. I'll just type out the command. But for things like this, if you're like, you don't want to be sitting there watching everyone or having everyone watch you make typos, it can be nice to have that. Yeah, I'm happy to, if there's any more questions about anything today or yesterday. Oh yeah, got one.
So you emphasized this earlier of the global motion from as soon as the ligand is bound to the FaNaC1, from ligand bound all the way down to the transmembrane domain. Have you like implemented the tools to explore the motions between the two states that is available on CryoSPARC?
So we will talk about some techniques today that are more focused on that kind of continuous deformation, right? Things like 3D classification are like yes, no, A, B. In the next case study, we'll talk about 3D variability analysis and 3D flex, which are more like kind of thinking about these sort of continuous deformations that happen. But also, I'll kind of say that when you're looking at a map and you see motion of the map, it can be hard to know sort of how much is like a rigid body movement versus how much is an amino acid actually moving kind of relative to its neighborhood. And that's the kind of thing that I think you're better served by building models and refining models into a couple states and then doing some kind of interpolation of the models. I've found that more helpful in terms of understanding these kind of continuous deformations that you see, and maybe some biochemistry that you see as well, if your map provides for that.
So then what would like qualify, say, for something like this, beyond just like the atomic modeling that you do, that makes you think, okay, maybe it's time to move on to 3D variability analysis or 3D flex?
That's a really good question. So you're not going to like this answer, but I would always just do both. I think in biology, we know, right, this is not, this ion channel is not just snapping closed, right? There's always going to be some kind of translation between these two states. And the question is, are there enough particle images in that transition that we can actually make a map of it? Okay, and we'll talk about that in the next section. But remember that when we're looking at these maps, we're looking at like ensembles of many, many particles. And so we know, or we can be confident, that we have a lot of particles that are in the apo state. We have a lot of particles that are in the ligand-bound state. The question is, when we make a map that is somewhere halfway between, right? Maybe the ligand is nearby and the thumb is halfway closed. How many particle images really are in that conformation? Okay, and so that's where we start to get into this tricky bit and we're bleeding into what we'll talk about next, which is fine. But that's where we get into this tricky situation with things like continuous heterogeneity, is that you never know when you're looking at a map, like how much support from the data does this have? And we'll talk about, we have ways that we can be sure we're not totally making things up. But in terms of like, when would I do one or the other? I would try both, and then I would really investigate if I just take particle images that I have modeled as having this kind of conformational state in the middle. And if I take those images and just reconstruct those, kind of what does the map look like, that kind of thing. So I think, yeah, and then
to that a little bit, sorry, just to add to that a little bit, I think everything that Richard said is correct. You can't actually ever know if your particle absolutely has discrete heterogeneity or continuous heterogeneity, and those definitions of those are what really Rich mentioned before. It really comes down to a question of like the energy landscape of this particle. If the energy landscape is very tight around two different states and there's very low probability of finding a particle in the middle, that is what we would call discrete heterogeneity for the cryo-EM context. Whereas, of course, in practice, the particle itself is not a discrete object ever, the protein itself. But for the purpose of cryo-EM computation, we have to make that assumption. Continuous heterogeneity for cryo-EM really means that the energy landscape has a shallow region where there is a probability of sampling a particle along that region. And so that's really the question you have to ask, is like, do you have any evidence to suggest that the energy landscape is shallow somehow, maybe from some other method, not from the images themselves? Or do you have some other knowledge about this particular ion channel or family or whatever else that may suggest that you definitely have discrete or definitely have continuous heterogeneity that you expect to see in the images? At the end of the day, another thing to keep in mind about all of this is that for refinements of an individual particle, so this is like homogeneous refinement and what we talked about yesterday, the validation tools are relatively well built up. So gold standard FSC is trusted amongst the community as a way, if you haven't broken any of the assumptions, a way to tell that your data is self-consistent, that you can trust your results up to a certain resolution, etc. There isn't actually an analog of that for heterogeneity at all. There is no method that is accepted by the community or really used in practice, even just for something as simple as 3D classification or heterogeneous refinement, to tell you that like you have some statistical validation that these states are present and that you haven't missed anything. The way we try to give confidence around these results is to make sure that we're not bringing in bias from any other outside source. So we're always trying to use the data only as much as possible. Our initial volumes going into these jobs are trying to be as unbiased as possible, often like we see in this example, using the same volume and letting the data separate into different classes rather than providing an open and closed version of the channel or anything like that as initial models. With all those safeguards in place, when you run these jobs, you also have to keep in mind that there is a possibility, especially with the discrete classification tools, and this is a fundamental problem of clustering methods, there's a possibility that you've missed a state. Like there could be a set of particles, some subset of particles that produce, if you were to identify those particles, those would produce a reconstruction of a state that you haven't seen. There's always that possibility. And I think the example with the yellow and green map, where doing heterogeneous refinement, this one, like in the way the story was presented and the actual order that this processing was done, we already knew at this point that we expect to find two different versions of the like thumb position. But if you didn't know that and this was your first job that you ran, then you would look at this and be like, you should look at this and be like, okay, there's clearly some broken particles or something not right about these particles, but I actually don't know for sure whether this class, even though the map looks good, is that actually homogeneous, or is there still another subpopulation in here that I just haven't identified yet? And this is a fundamental question about clustering methods. And this is like not even cryo-EM specific, just generally speaking, any method that clusters data, clustering is fundamentally a difficult problem, and fundamentally a problem that's not guaranteed to give you the same solution every time you try to run it, heavily dependent on initialization and other randomness that's part of these algorithms. So it's recommended also, this is just sort of an aside, that for extra certainty, you can try running the same jobs multiple times. There's a parameter in CryoSPARC called the random seed, and by default, it changes every time you run the job. So if you just clone a job and running again, you will get different results potentially, and that's also something worth trying just to be sure. I mean, not sure, but more sure than just running the job once that you haven't missed something. So these are just some extra thoughts around.
So the kind of, just because this was a very long tangent, sorry. So the takeaway that I'm getting from this seems to be, one, there is sort of an unexpected state that is very well populated that would kind of lend itself to, okay, maybe we can explore this sort of heterogeneity a bit more. And then the second bit would be, we have auxiliary techniques that will back up that sort of intermediary technique as of now. Am I kind of?
Yeah, I think that's mostly right. And then I also think, like I said, I think you can try both. And then when you look at, for instance, if, when you run 3D-VA, you'll get 3D variability analysis. We'll give you like a distribution of your particles along the kind of like coordinate, right? And we'll talk about this more later. But if you see that it's basically all the particles are in this Gaussian peak or this Gaussian peak, and there's not really much in between, you can say, okay, based on the particles I picked and the specifics of this job, it seems like it's more discrete. Or if there is kind of one big Gaussian that's like spread across the coordinate, okay, maybe it seems more variable. But then I think, like Ali said, and like you mentioned as well, you can't ever be certain. And so, the other techniques that, like, maybe something like FRET or something is telling you that you have the switching between these states is very slow, or maybe it's very fast. I don't know. I'm a bad biochemist. But, that kind of thing, like an orthogonal way to verify the types of heterogeneity you're seeing in cryo-EM is always going to make you more sure, right? Because these clustering methods, just mathematically, you can't ever be sure that you found everything.
While we're on this, I'm just going to interject with one Zoom question that's very heavily related to this. So the person is asking, in this case, let's say the hollow form is quite rigid. So you have one 3D class that might absorb all the particles for that. But that the apo form has two distinct sources of heterogeneity. So potentially partial occupancy, but then also continuous flexibility. And so basically what they're asking is, what approach would you want to try? Could you, for example, try a 3D classification with many classes to capture all the states of the apo particles? And I think this is going to be covered quite a lot, I think a little bit in the next session. But if there's anything you wanted to touch on about strategies now, we could maybe do that.
Yeah, I think that's a really good question, is like what, sort of building on, like you say, like, we can't ever be sure we found everything. It's like, well, then when do you stop? And I think it depends on what you're trying to do, right? In this case, what we want is to get really good maps of both of the states and really try and characterize the ligand binding as much as we possibly can. And because of that, I think if I were a grad student and this were my project, I would be sitting at the computer and like, okay, I found this class, like let's run another heterogeneous refinement. Okay, I ran another heterogeneous refinement. I found these two classes, but maybe as you say, okay, the TMD down here, why is it still blurry? And it hasn't moved very much, like your question was, right? It's a little different, but it's not as different as I'd expect. Why is that? Is there more heterogeneity in there? Maybe I'll try another heterogeneous refinement. Maybe I'll take each of these classes separately and run some continuous heterogeneity jobs to measure that, right? I think you can go, you can go as deep as you want and as wide as you want, right? Maybe you repeat, okay, I did heterogeneous refinement. Now I'm going to do 3D classification, see if there's different numbers of ligands bound, right? Maybe the reason the TMD is still blurry is that we have like non-stoichiometric binding, right? And I think if your project is characterize the effect of FMRFA binding on FaNaC, you're going to spend a year or more processing just this data set to try and get as much as you can out of it. If your project is, hey, is FMRF binding our sample? And you get a map where it's bound and a map where it's not, you're like, yeah, and then you're done, right? So I think, and that's just my philosophy, right? There are certainly people in the field of cryo-EM, and I'm not saying they're wrong, who are like, it is a disservice to the community if you don't push your data set as hard as you can push it. Because let's be honest, you upload this to Empire, please always upload your data to Empire for us. You upload this data to Empire, maybe 3 people download it, right? And one of them is me. And you've seen what I do. So, no one is ever going to know your data better than you. You are going to spend so much more time looking at your data than anyone else. And so if you leave something on the table, it's probably going to be on the table forever. That's not to say you need to do a perfect job, always going as deep as you can into your data set, but you can go really far. So it's this trade-off, right? And it's the question of your time. I think, how do you know there's more? There's probably always more you could find. The question is it going to be, if I spend another month working on this, I find another cool state, or like I think that's what's going to happen? Or is it going to be if I spend a year, I get from 2.7 to 2.6 angstroms, right? That's the kind of things I'm kind of talking about.
Yeah, I think that's a really good point. And just to add to that again, I think there's advice always about how much data should you collect, how much time should you spend on a project. And I think what Rich said is really right. It really comes down to what your goal is. I think one thing to keep in mind is that for projects where your goal is just high resolution structure determination of a single state, you're looking at the first structure of something, people often suggest that you don't really need that much data. If you're collecting like tens of thousands of movies, you're probably overdoing it. It's better that you focus on optimizing your sample, collecting less data of higher quality, etc. Heterogeneity is a case where the opposite can sometimes be true. Not to say that you should ever collect poor quality data, but if you imagine even a data set like this, which is in terms of heterogeneity that people see in real life, you could even call it simple compared to how many states or how many classes some proteins have, some systems, whatever you're trying to study. Every time you do one of these steps, the numbers of particles you have in each subdivided bin shrink further and further. And sometimes you get to a point where you can see, like right now, you can see that there's something up at the bottom of this map. This is not done yet. But if you only had 10,000 particles at this point, you're kind of stuck. Like you can't really go further. So it's worth considering also that if your goals really are to explore heterogeneity of a system like this, you should first optimize your sample so you can get a really good high quality single structure of the most populated state to make sure that you know how to do the sample prep, you know how to do the imaging, and everything is good. And then you should actually collect lots of data and you will spend a lot of time processing, but you can uncover some really amazing things. And I think that's the reason why heterogeneity, generally speaking, is still the really big open frontier for cryo-EM. Methods are still being developed, techniques, strategies, everything is still in flux. And like I said, validation is still TBC. So it's an exciting area. Yeah.
What has been said. And so we have solved many structures. And I'm pretty sure the structure we published were not wrong. But did we get everything out of it? I'm pretty sure we have not. So as a result of that, I really encourage the community to deposit your data to Empire. I try to encourage NIH to set up a server or some mechanisms, but at least some years ago they said, oh, it's so easy to get data, you just get it again. But actually, that's what they set up is S2C2 for you to get that again, which I like. On the other hand, I think there will be really interesting questions. You see, when I'm sitting here, the question being asked is exactly came to my mind. You have one way, but I go another way. Which way is better? Depends on what you want to squeeze out of your data. So as we speak, we actually, people in the lab are repurposing some of the old data with new software available that we heard today and tomorrow. So I think it's extremely interesting. And I think in the long run, it's a good thing. It's just like the AlphaFold. Thanks very much, PDB, for the last 60 years for people to deposit the data. But if you don't have that data, they will never have an AlphaFold, in my opinion, okay? So I think with this kind of data we have collected, I think if we can upload that in the Empire, I think extremely useful for either you process, which is hard to measure the impact because you use an example to teach more than 200 people at one time. Is, you know, you're not publishing a Nature paper in your talk, right? So in the normal academic, you know, you're nobody in a sense, but for us, it's a hero because you teach us exactly what to think about. So I think it's a continuing discussion with the funding agencies. Now, in Europe, it's different. In Europe, everything has to be deposited no matter what you do. Okay. So in American, we don't. They said, okay, just the map. And even the map is challenging, right? Which map you deposit in the EMDB? The one on the left, the one on the right, or both, or what? How about the junk map? I find the junk map actually interesting because that could be, if you refine your green map again, if you have time, I encourage you to do that, you may squeeze out something really different. And I'm thinking about, from a mechanistic point of view, it's really not looking at the local region, all the allosteric interactions and so on. That's equally important, particularly you do a drug design.
Yeah, and just to add to the, I think that's a really excellent point. And just to add to it, especially from our sort of maybe more selfish point of view, believe it or not, Structura does not have a Titan Krios. We don't purify protein and collect data, right? We rely and really need data from the community, from places like SLAC and other places with microscopes, so that we can access data sets that help us develop techniques like this, right? We need cryo-EM data to develop the processes that work on cryo-EM data, and we can't generate that data. So we really do. It's kind of a symbiotic relationship, right? We develop techniques for the community, but the community lets us develop those techniques. And something we talk about a lot is that because of how depositions work, we have access to a lot of good data. We have access to almost no bad data. And almost no, and when I say bad, I mean, we don't want just a bunch of ethane, but bad being hard. Like I couldn't get a 2.5 angstrom map out of this, so I didn't publish it. That's the kind of data that I think we really crave, right? So, you know, I think anytime you can convince Empire to take your data, I think that we'd love it if you put it there.
One more thing, and going back to the original design of this experiment, if there was one grid that was APO, and you solved the APO structure, you would answer a lot of these questions about the relationship of APO to the bound form.
Yeah.
Where here you've mixed them, and you're depending on CryoSPARC to tell you where is the APO. Doing a structure, designing the experiment, do the control.
Right. That's okay. So the question was, what's the particle number in this map? I actually don't remember, unfortunately. Certainly fewer than 1 million and more than 100,000, but that's the order of magnitude. I'm sorry, I forget for this exact job. Sorry.
Hi, there's a question we didn't discuss much, but I'm curious, this denoiser you used, because my impression is denoise micrographs is usually to facilitate manual labeling for further training of a deep picker, because you lose a lot of high resolution information. Is it good for because do you also use a denoise image for template matching, or is there any cautions to take when you feed the auto picker with denoise images?
Yeah, so that's a good question. So if you look, it is true. We've lost basically all the high resolution information, and I'm not even sure it's meaningful to think about it that way, because we basically, I mean, the denoised image is just fake, right? It's real in the sense that it came from an image we collected, but like the signal in the denoised image is something that has been sort of amplified using this nonlinear process that, you know, I wouldn't make any claims to say we've just lost high resolution information. We've done a lot of things that we don't even know what the model is doing, right? So that's kind of the first part. The second part, what do we use it for? In this case, I blob picked and template picked on the denoised image. And we have found that basically that increase in contrast, even though kind of traditionally you would use these denoised images just for like deep pickers, things like Topaz or Cryolo, for instance, we have found that blob picking and template picking also benefit. So I recommend doing it basically from the get-go. The main difference is that the denoised image for template picking, you don't have to think about CTF. The denoised image has already kind of had the CTF baked in, but CryoSPARC handles that for you automatically. If you turn on the use denoised image for picking, it will also turn on the setting to ignore the CTF. So it's all kind of baked into the thing.
I'll talk about this topic a little later today as well, about how the denoiser works and why we think it's really useful for particle picking. But I think in our hands so far, on every challenging data set so far, we found that denoising and then blob picking or template picking on the denoised micrographs works really well, and especially for avoiding empty ice regions or particles that are picked on contaminants, because the contrast, not only is the contrast enhanced by the machine learning model, the grayscale level of the particles becomes a lot more consistent, and I'll talk about this later as well. And so across a range of defocus values, like this is just a single micrograph at a single defocus value, but you can imagine another micrograph from this data set that was collected at much more or much less defocus. It's actually going to visually look very different. And that often throws off particle picking as well. Denoising also takes care of that problem. So we'll talk about this a bit later today.
Okay, and then I started way ahead and now we're behind. So it's time for a quick break. Sorry, Amy. And then, yeah.